Hi, this is Miss Slitton, and these are some members, though few, of AP Biology, and we're going to go through the Unit 3 essays real quick and make sure we understand them. The first one that is listed is all new cells come from previously existing cells. The process of cell division, which involves both replication of the cell's nucleus and division of the cytoplasm. Okay, so tell me before I go any farther. When we say replication of the nucleus and division of the cytoplasm, what do we call division of the cytoplasm? Cytokinesis. Good. And then when we replicate and divide, when we divide the nucleus, that's called mitosis. Good. To form new cells. There are two types of nuclear division, mitosis and meiosis. So on A, name and describe each stage of mitosis and then do the stages of meiosis. Now, it says on that part, what I would highly, oh, sorry, I had this part frozen. I am recording it, but you can't see what I'm doing. My bad. There you go. So when you do this, you want to be able to replicate the stages of mitosis and show me with chromosomes. Okay, so for instance, you might say, okay, here's a cell, and this is what it looks like during interphase. Okay, and then you could tell me, you know, if I tell you that, let's say the 2n number is 6, okay, how many chromosomes are in that cell? Total. How many chromosomes are in that cell? 2n number is 6. Then there are 6 chromosomes, right? If we're going to do mitosis, then we need to make 2 cells, each that have how many chromosomes in them? 6. If we were going to do meiosis, we'll make how many cells? 4. And how many chromosomes will be in each? 3. Good job. Okay, so you would say to me that if I looked inside of that nucleus, I would see 1, 2, 3, 4, 5, 6 chromosomes. Do you agree? Okay, and if we are going to do prophase, that's when the chromosomes become what? Visible, and the nuclear envelope starts to break down, right? On a metaphase, you would see something like this. Yes? And I would see microtubules, spindle fibers coming across here on a metaphase. And then on an anaphase, I would see something like this. 1, 1, 2, 2, 3, 3, 4. Yes? And I would show maybe some spindle fibers pulling them apart. And then on telophase, cytokinesis, I would end up seeing 2 cells, and each of them would have 6 chromosomes in them. But now each of those chromosomes would appear as a single, right? And the only reason they appeared double right here is because we did DNA replication. And in what stage do we do that? As stage of interphase. Now, if you were going to do meiosis, this would look different, right? You would take your 6 chromosomes, and when they became visible, we would see them like this. And this would be an example of what? Metaphase what? 1. And then as we move to anaphase 1, okay, it would look something like this. Right? And then telophase 1, you would divide these into 2 cells, and then anaphase 2, we would separate the sister chromatids. So you would be able to describe what is going on in each of those steps as you work your way through. Okay? Now, the second part of that question says, the first part says, name and describe each stage of mitosis and meiosis. The second part is, please describe the importance or role of meiosis in gamete formation and variation. So what did we say about meiosis on ways that we have variation? Where are the areas where we get variation? Crossing over. And you would tell me that crossing over occurs when? Prophase. Yeah, crossing over occurs during prophase what? 1. Where's another place of variation?
Independent Assortment.
Yes, independent assortment is another place. And this is because these homologous pairs can line up any which way into the other one, and that would happen during metaphase 1. Okay? And then the third way it contributes to variety is all the cells that you make, they're all going to be what kind of cells? Haploid cells, right? And because you made haploid cells, you need to have two haploid cells that get together in order to form a diploid zygote, and you don't know which haploid is going to get together, so that's the way you would have variety. Okay, does that make sense? 3 areas for variety. Okay, so let's look at the next question you have. Cancer is the failure of genetic control. What are the general characteristics of a cancer cell? You can look those up. I gave you that, and you have a chart, characteristics of cancer cells. Okay? Then B, it says, what two types of genes in particular malfunction in cancer cells? What are those two types of genes? We called them. What do we call them?
Tumor suppressor.
Yeah. Tumor suppressor and what? And oncogenes. Okay? So they were proto-oncogenes before they became oncogenes, right? And we said oncogenes are facilitating the cell cycle. And an example of an oncogene we said was RAS. Remember we said RAS is like stepping on the gas? And it is coding for proteins that are facilitating you moving forward in the cell cycle. That's not great, but hopefully your tumor suppressor genes like who? P53, who codes for P53 proteins or caspases, those go out and kill, okay? If they're working well, that'll kill the RAS, right? Those will kill the cells that are out of control. But if they're not working well, it's a combination of cells that are going and replicating too many times and nobody telling them stop. It's a combination of those two things that actually give you cancer. So what you would want to do is, what are the two types of genes? You would name them, and what do these genes do in normal cells? Normally, they're in positions of control. A proto-oncogene will stimulate the cell to undergo the cell cycle or not, and normally it functions just fine. But when it has mutated, that's when you have a problem and it becomes an oncogene. Normally, tumor suppressor cells work just fine. P53 genes code for proteins that stop the cycle, allow the cell cycle to repair itself, and if it doesn't repair itself, then they what? Kill the cells. So when those aren't doing their job, that's when you end up having cancer. All right? So let's move on to our next question. It says, state the conclusions reached by Mendel in his work on the inheritance of characteristics, and then explain how the following deviate. So the first thing you have to do is give me Mendel's conclusions. So what are Mendel's conclusions? The law of dominance, the law of segregation, and the law of independent assortment. So you would explain that. A dominant trait can hide a recessive trait. You would talk about segregation, that homologous pairs separate, and that you only get one copy of the two alleles that you have. Only one copy of those alleles gets passed on into your gametes, right? Either the one that came from your father or the one that came from your mother. And the law of independent assortment says we can have any combination of genes on your different chromosomes depending on the way they line up. Unless those chromosomes are what? Unless those genes are what? Linked, right? Okay, so let's talk about that. Here's the first one, autosomal linkage. Okay, because this is saying, explain how each of the following deviates from these conclusions. So which one does autosomal linkage deviate from? Does that affect the law of dominance? No, traits are still dominant. Does it affect the law of segregation? No, you still only get one copy. What does it vary from? Law of independent assortment, exactly. Law of independent assortment is who that doesn't work with, because that's saying I don't get to assort independently, right, at metaphase 1. And if I have different things that are on here, okay, I could get some of these black with the green dominant recessive, right? It doesn't happen. But because they're linked, because they're on the same chromatid, they travel together. I don't get independent assortment. They're going together. What's the only thing that breaks up linkage? Crossing over. Okay, so now let's look at the next one. Sex-linked inheritance. Okay, which one does that not follow? Sex-linked inheritance, which one? Does it follow the law of dominance? Does it always? Because the law of dominance says in order to see a recessive trait, you need 2 copies, right? That means you have to get a copy from both of your parents in order to express a recessive trait, right? But you don't right? Because a guy gets how many copies? 1. But yet he's still expressing it because he only gets 1 copy. So that would be an exception. Sex-linked traits would be an exception. I don't know where I was going. Sex-linked traits would be an exception to dominance. Yes, dominant traits are still dominant over recessive traits, but in order to get a recessive trait, you only need to get 1 bad copy and you will get it. Where we are used to having to get 2 of those alleles in order to get that particular trait. You all right? Question on that one? Okay. And if you look at sex-linked, we're still segregating. You're still only getting one or the other. And independent assortment, we're not really talking about that because we're just talking about the genes that are on the X chromosome. All right? Polygenic, multiple gene inheritance. Okay? What one is that talking about? What one would you say? Are you still doing segregation? Yeah, you're still separating. The only time you don't follow through with segregation is when you do non-disjunction, failure to separate, meiosis. That's going to cause a problem. So there's nothing that naturally happens that way. That would be an abnormality. Okay? Are you still doing independent assortment? Yeah, you can still assort independently, but this would be, again, on your law of dominance. And the reason is, we said before, you have to get, as long as you get 1 dominant trait, or 1 dominant gene, you're going to get that. In this case, remember we said in polygenic inheritance that you're going to have several different combinations in order to get that 1 trait, and the environment can affect them whether or not you get that 1 trait. It's not simple dominance. Okay? It's not simple dominance. Okay, can I move on from that one? All right, next one. It says, discuss the variety of gene interactions listed below. Be sure to explain the inheritance pattern, should be an N, as well as give an authentic example of each. Incomplete or partial dominance. So for incomplete or partial dominance, you would say 1 allele isn't completely dominant over the other, right? In our eyes, it looks like what? What does it look like in our eyes when we have incomplete dominance? It looks like blending, right? That was red flower crossed with white flower gives you pink flower. That's straight hair person crossed with curly and you get a wavy. That's an example, an authentic example of incomplete or partial dominance. Epistasis, when we talked about epistasis, we said 1 allele controls another whole set of alleles and the way they are expressed. If you remember for that one, we talked about rats, right? And rat color, you could either have a black rat or a brown rat, but you're going to have an albino rat if you don't have the big C, the epistatic, right, gene. Because if it was big B anything with a big C, you got a black rat. If you got 2 little b's with any big C, you got a brown rat. And it didn't matter if you got big B's or little b's, if you didn't have a big C, you were going to be an albino rat. So that C gene controlled the laying down of the pigment, whether or not that pigment coded for black or brown on your pair of alleles. Okay? Pleiotropy. Remember, that was when we said you could have 1 gene here that codes for several different, seemingly different traits. And we talked about what on that? Sickle cell, what? Sickle cell or any kind of syndrome, right? We talked about where you get several different results. And we talked even about sickle cell disease and it giving you greater resistance to what? Malaria. Okay. Multiple genes or polygenic inheritance. We've already talked about that. That's when several different genes contribute to that trait. An example we talked about there was intelligence or height or skin color, right? That was a polygenic trait. Yes?
So it's like multiple genes relating to just 1 trait.
Yeah, whereas here we have 1 gene giving, yeah, it's like the opposite. If you go this way, These are several different genes giving you one trait. Exactly. Okay, and then multiple alleles, if you remember for that one, we talked about blood type. There wasn't just big I and little I, there was big IA and big IB, and you can get any combination of those alleles. You still only get two, but it's not like we just have a dominant and a recessive, okay? We have choices on our dominance. And then we talked about how this was an example then of codominance, because both are expressed. Remember that? You get the A antigens and the B antigens. All right? Okay, moving on. Looking at your next question. Experiments by the following scientists provided critical information concerning DNA. Briefly describe how each contributed. We haven't talked about these yet, so we can't review this question, okay? But we will talk about these three, okay? And what was the next one? The structure and replication of DNA, we haven't talked about. You know the structure of DNA. You know that DNA is made out of what? Nucleotides, and that's a what? Base, sugar, and phosphate. But these two essay questions, what was this one? These three essay questions all come out of chapter 12. So when we're going through chapter 12, let's simultaneously address these essays as we go through them. Okay, so I got through everything in stupendously quick time. Are there any of these that you want to go back to talk about from the beginning? Is there any of these that you want to have a little more discussion about? Just kind of running through these. You okay with all that?
Wait, this one is just explaining each of them and giving you examples.
Yeah. So that I know you have an authentic example of, oh, I heard there's epistatic genes. What does that mean? That you can tell me what it is in words and then give me an example to show how that is a model of epistasis. Yes?
What was, I think you said it, I just forgot. What was one example of the polygenic inheritance?
Polygenic, that would be things like skin color. Remember how we looked at the A, B, C genes on that one? Or I think we just looked at A and B on those. Or wheat varieties or height. You know, you have that bell-shaped curve. Okay, that was that polygenic. Or sorry, yes.
Does blood type count as polygenic?
It doesn't. That's multiple alleles. You're still just dealing with eyes, right?
So
Variation on the eye. Just the single homologous pair. You have lots of variations there. Polygenic is I's, R's, Q's, P's, and L's, giving you your trait. Okay, what else? Yes?
So that they're giving you the trait, what trait would that be? Like skin color?
Yes. What else? Okay, so I think that gives you a good starting point. Get those done, and then we can work on these next three as we're going through the chapter next week. Because the next three are basically the chapter, the summary of the chapter. More? You good? Okay. All right, that's it. Good reviewing. And if you're staying up really late, eat a piece of toast. Okay, I love you.